Good morning. Same as always, locked door, shut, empty room, just me and the computer. Chapter 10, Biotechnology. Again, Biology 1300. Oh, I guess it would help if I actually turned this thing on. There we go. All right, so about 99% of your DNA is exactly the same as everyone else's in the room. Well, there's no one else in the room, but normally I have a room full of people, so I say 99% of yours is the same as everyone else in the room. A nucleotide difference carried by at least 1% of the population is a single nucleotide polymorphism, or SNP. Differences in alleles are the basics of the variation in human traits that makes each individual unique. By differences in alleles, again, we're talking about you have genes for eye color, and then the alleles would be brown, blue, green, the other eye colors that exist. Personal genetic testing may allow physicians to customize treatments on the basis of an individual's genetic makeup. From the time this textbook was written several years ago to now, there's been a lot of new, what is called genetic therapies. We are doing gene therapies. We're testing different, excuse me just one moment. We're testing different ideas and different ways to treat different diseases based on a person's individual genetics. We can actually engineer cancer treatments to attack only cancer cells. It's in the early stages of that. They're doing some of that work over at UAMS in Little Rock and other places as well. But it's one of those things that hopefully in your lifetime, maybe not mine, but in yours, this will be a way that we can get a much better handle on treating things like cancer without using radiation and chemicals and all that. We can engineer a virus to actually go after your cancer cells and help you that way. This is everybody I think knows. This is Angelina Jolie. She was tested. She had a family history of breast cancer. She was tested to see if she might have what is referred to as the BRCA1, or is it BRC1? Anyway, it is a mutation of a particular gene that in about 30 to 40% of cases leads to breast cancer. She discovered that she was positive for that mutation. And as a preventative measure, she underwent a double mastectomy and reduced her risk of developing breast cancer from about 80% to 5%. And she did that through, again, this is her individual testing of her DNA showing that she had the mutation. She went through the double mastectomy to, again, reduce her risk of breast cancer from something like 80% down to 5%. Some other testing. Results often include predicted likelihoods of having traits associated with particular SNPs. Alzheimer's disease, everybody pretty much knows or has seen something to do with Alzheimer's. The lifetime risk of developing this is associated with a gene called the E4 gene. People with this allele have about a 30% lifetime risk of developing the disease. That means that 3 out of 10 people with the allele eventually get the disease. Here's the issue with our genetic testing currently. Many people without the allele develop Alzheimer's. So this is not, again, we get real careful in biology when we start talking about cause and effect. This is the effect. This is what causes it. This is not the cause of Alzheimer's. Because if it was the cause, nobody that didn't have that gene would get it. This is what we refer to as a contributing factor. So again, if you were tested and you saw that you have this gene, you would know and possibly be able to prepare earlier for the onset of Alzheimer's disease. Gene research traditionally was limited because of just our technology. But once we discovered restriction enzymes, we began to be able to do much more with gene manipulation and following genes and tracing genes. Restriction enzymes are produced by bacteria. These enzymes cut DNA at a very specific place. and they are used in DNA cloning to cut DNA into pieces that are then inserted into what are referred to as cloning vectors. A cloning vector is an item that will clone the DNA, make multiple copies of a particular gene. When used appropriately and using the right restriction enzymes and certain DNA ligases, researchers can cut and paste DNA from different sources. You can take DNA from one animal and place it into another animal. You can take it from one plant and put it to another plant. You can take it from an animal and put it into a bacteria. You can do all kinds of things. We now have the capability using DNA ligase and restriction enzymes. The result of that modification of DNA is referred to as recombinant DNA. The recombinant DNA is a hybrid molecule of DNA from two or more organisms. Again, there used to be ways we could manipulate things by, like in plants, we could cross-pollinate things, and in animals, we could try to crossbreed them. It didn't always work, but through this use, it's much easier and a much more efficient way of developing this recombinant DNA. That's the first step in DNA cloning. There is a set of laboratory methods to use living cells to mass produce specific DNA fragments. One of the things we can produce this way now is insulin for diabetics, specifically type 1 diabetics. Historically, a type 1 diabetic that was insulin dependent, that had to get their insulin, they had to inject themselves. We all know those. We've heard of people who are type 1 diabetics and have to inject themselves, before they eat or after they eat or however their blood sugar is running. These people when they had to buy their insulin from the pharmacy, that insulin had to come from somewhere. Now, unlike blood, which human beings can donate, I can go down and give, a pint of my blood to the Red Cross, and the Red Cross can then sell it to hospitals or whatever they do with it to get it in the blood supply. Insulin's different. We don't need insulin, we don't have insulin. We don't make it until we need it. So we don't have an excess supply. So if I was to donate my insulin, then I would become a diabetic. So where did the insulin come from that people were buying to inject? Think about it for a moment, and I'll tell you, it came from pigs. The insulin that was used historically came from pigs, and it wasn't donated. It came from the slaughterhouse. When the pigs were killed to make ham and pork chops and bacon, the pancreas of the pig would be collected, and then the pancreas would go through a process. I have no idea how it was done, but they would process it and they would remove the insulin from the pancreas. Now, this is a very labor-intensive way to do things. There was a really high risk of contamination with all kinds of issues, but that's where it had to come from. In addition to that, there's also several groups of people that might have a real problem using a porcine or a swine product. Think about our Jewish friends and our Muslim friends, all of those religious groups or any other group, maybe a vegetarian or a vegan may say, I don't want anything that a pig had to die for. So that was something, that had to be considered when you were going to take this medicine. Were you going to use this product that came from pigs? We don't have to get it that way anymore. Through biotechnology and through the use of recombinant DNA, we can now make human insulin in the lab. Now, are these porcine products still available? Yes, they are. And some people, if you've been a diabetic for 20 years and you've always used this particular insulin and it works very well and it does a really good job of controlling your blood sugar, maybe you and your doctor don't want to change. You want to keep using what you're using. But if you're a newly diagnosed diabetic and if this is the way you want to go, we've got options. You have options now that we didn't used to have. And we have those thanks to recombinant DNA. Making recombinant DNA, the restriction enzyme EcoR1 recognizes the nucleotide sequence GAATTC. Don't worry, I will never ask you to remember those letters. And from two sources, you see here's the first source and here's the second source, but it recognizes GAATTC. See the same thing. And if you'll notice that I'm sure you recognize that the opposite sequence is going to be the same on each one of these different. Now all of this information is different from this information, but this little segment right here is the same. And the restriction enzyme is going to break all of those bonds and cut that at that particular place. The enzyme cuts the DNA into two fragments, and we refer to these little ends here as sticky ends. Now, since these bases are the opposite of these bases, this can now stick here, and this can now stick there. Those reactions or those binding spaces will happen. That's why they're called sticky ends. They will stick there. You see now we've got the red and the blue together here, the blue and the red together here. So we have now DNA fragments from two sources are being mixed together, matching their sticky end base pairs there, and we now have a piece of recombinant DNA. A DNA molecule that can accept foreign DNA is referred to as a cloning vector. Lots of cloning vectors out there. We're not going to go into great detail about any of them specifically. If you continue biology at later in life and maybe you take a genetics class, you'll talk about lots of different cloning vectors. The one cloning vector we are going to talk about is something called a plasmid. A plasmid is a small piece of DNA found in certain bacteria, and the value of the plasmid is that it will replicate outside the normal replication of the DNA. It replicates independently of the chromosome in the DNA of that bacteria. So plasmids are often used as a cloning vector. As a matter of fact, at this time currently, they are the #1 cloning vector used worldwide in biotechnology research. Now, as again our technology improves, something may overtake it later, but right now it is the most oft used of the cloning vectors. And again, it's a small circular piece of DNA that's found in bacteria. Here is an example of how cloning works. Here's the normal DNA. This is the chromosomal DNA, and here's the cloning vector. So what we will do is we quote the we here, humankind, science, recognizes a place where a restriction enzyme can cut, creating those sticky ends, and it recognizes it here. We have the restriction enzyme, which is the gold triangles, cuts a specific nucleotide sequence in the chromosomal DNA. So now you've got these little pieces here. These are the little chromosomal DNAs. These are the fragments. Let's say this is a gene for the production of insulin. This is the gene that produces insulin. We take this plasmid, we cut it open here, we insert the gene right here. Now normally then the recombinant DNA is inserted into a host bacterial cell, like say E. coli, and we use E. coli a lot because it reproduces very quickly. We can make lots of copies in a hurry. Now understand that once this cell gets in here, normally, when this plasmid starts to reproduce, all it's going to produce is another plasmid. But by placing this gene here into the recombinant DNA, when this plasmid starts to reproduce, not only is it reproducing all the blue stuff, it's also going to reproduce the red stuff and the product of this gene. And I've already said the product of that gene in my example is insulin. So here you place it in here, and then you've got all these subsequent generations of the bacteria producing the plasmid. DNA libraries is where we collect our DNA material. It is a complete set of an organism's genetic material. There is a DNA library for human beings. There's a DNA library for foxes. There's a DNA library for rose bushes. All different organisms have DNA library. But it's a collection of cells that have different clone fragments of DNA. The fragments of DNA or RNA can be labeled with a tracer or a probe. That way, by certain radiography or X-ray technology, we can actually see the inserted gene, the recombinant DNA. And it can be used to find a particular clone containing a desired sequence. The probe and the gene are complementary, so the two can base pair, which is called nucleic acid hybridization. For you to be able to place a gene in a new organism, there has to be a place that is compatible or complementary. And again, this process is called nucleic acid hybridization. If you are a fan of the TV shows about crime, you are familiar with the phrase polymerase chain reaction. Anytime, especially 10 to 15 years ago, when they had those crime shows and they would talk about matching DNA, they would always say using a polymerase chain reaction that the DNA was modified. This is a technique used to rapidly generate many copies of a DNA fragment without having to clone it in living cells. It's based on DNA replication, uses primers and heat-resistant DNA polymerase. I did some of this when I was at UALR as a graduate student, and it's really kind of cool. The machine that is used for PCR actually kind of looks like a toaster. You know, a toaster oven that has a little drawer you drop down, you put your toast in there, or you can put some pizza pockets or, you know, any of those kind of things into one of those little toaster ovens. That's very similar to what a polymerase chain reaction machine is. It looks kind of like a toaster, but it's a really expensive looking toaster. And no, I don't think you can actually make toast in it, but it kind of does look like a toaster. A primer is a short single strand of DNA designed to hybridize with the DNA fragment to pinpoint the desired sequence to be amplified. A primer is something that primes it. If you've ever, used a pump, you have to prime the pump. You have to put a little water in the pump for the water to start pumping water forward. So it's a little piece of DNA that you put in to get this process started. 2 rounds of PCR. See, you're going to split the DNA right here. You're going to separate the DNA. You're going to put these little primers. These are the primers that get things started. And then you've got one going one direction, one going another, building this new piece of DNA. And again, this continuation, now we're going to split each one of these in half. And now we've got, here's the stuff, but now look what we have here. And what will happen in the third round, okay, I didn't think it would show that. In the third round, all of these would separate. And remember, this is the desired, this is what you're looking for, these segments here. So you're making this, and now you've got it doubled here and doubled here. Now this one is going to separate and make 4 more of these. This will make 4 more of these, and then we'll have this and this. So as you can see, as it goes, if you have round after round after round after round, see how much of this you will start to produce rather quickly. In the old days before PCR, again, I always go back to the crime shows because that's one of my guilty pleasures is I watch the crime shows to keep an eye on technology. My main purpose is I want to see how many mistakes they make with their terminology and how they say things. And I'll point out, I'll point to the TV and I'll say, that's wrong. And my wife will say, Tom, they cannot hear you. And I'll say, yeah, I know, but still I like to point out. But in the old days, you might have, say, a biological sample. Maybe you had a DNA sample or you had a blood sample from a crime scene. But the way our country's jurisprudence works, hang on just a moment. Sorry about that. The way our jurisprudence works is anything that the prosecution has, they have to give the defense to. They have to give it to the defense as well. So, because the defense has to have the ability to do their own testing on things. Well, in the old days, you may not have enough of a sample because, okay, I've got this much of a sample, but I'm going to destroy the entire sample doing my test as the prosecution. I don't have enough of a sample to give to the defense. So crimes would often go unprosecuted because while we have a little DNA or a little blood, we don't have enough. Well, thanks to this, that's no longer an issue. You take a little bitty sample and you just make a bucket full. And you say, here you go, Mr. Defense Attorney, here's all the stuff you want to test for this particular case. So again, this is how it plays its role in society. Sequencing of the DNA reveals the order of the nucleotides. That tells us the A's and C's and T's and G's. Sequencing produces millions of DNA fragments of different lengths, all incomplete copies of the starting DNA. Fragments are then separated by length using a process called electrophoresis. If you've ever seen those things on the TV shows where they show the thing looks kind of like a barcode, each one of those little bars are fragments of different lengths. They all start at one end of that gel, and electricity pulls them through the gel. The smaller fragments will move further in a given time frame than the larger fragments, but like-sized fragments bunch together, and they form those telltale bars that you see, on Maury Povich, when Maury Povich tells a certain gentleman, you are the father, or aren't the father. That's because the bars produced by that man's DNA and the bars produced by the baby's DNA have some common areas. Automated DNA sequencing and PCR enable scientists to sequence the more than 3 billion bases of the human genome. Human genome, very, very large, but again, almost all of it is exactly the same. Here's an example of DNA sequencing. And you see this is a very complex thing. I would hate to know that I had to try to pick out, if I had this one of one person, the father, for instance, and another one just like this of a baby, I'd hate to know I had to look and try to find which one was different. Private competition spurred a public collaboration to sequence the human genome. The Human Genome Project was undertaken by several different countries. The National Institutes of Health worked with some international institutes to sequence different segments of the genome. Basically, you had this organization working on this this area of the genome, this organization working on this one, and this organization working on this one, and together, once each one of those had done their job, then they could collaborate and put the whole thing together. Eventually, the United States, the UK, the United Kingdom declared that the gene sequence could not be patented, but the ongoing competition helped spur the genome to completion in 2003. So basically what these two countries decided was that if anybody sequenced the DNA, they couldn't patent it because if you patent something, nobody else can use it. And it was decided between these two countries that that should not occur, that the human genome should be free for everyone to use, all researchers to use equally. The study of genomes, or structural genomics, comparative genomics, variations in sequence. The study of genomes, what is it about the human genome that allows us to do things that other organisms cannot do? Why do we have the ability to, for instance, we can speak? What is it about our genome that it gives us the ability to speak, whereas other organisms can't speak? Provides insights into evolution and has many medical benefits. We have learned the function of many genes by studying their counterpart genes in other species. Yes, we share a lot of genes with other organisms. Other organisms have eyes that have color. For instance, dogs. Most dogs have brown eyes, but there are dogs with blue eyes. And there are lots of different shared genes. Pretty much every animal on the planet has a gene that's called the cytochrome C gene. So that's a gene that we all share. And what we look at in the comparison is how alike is my cytochrome C gene and a rose bush's cytochrome C gene. And yes, I know a rose bush is not an animal, but they still have the cytochrome C gene. Most living things on this planet have a cytochrome C gene. Again, genomic differences at different spots. Here's a gene, doesn't say what it is, but you can see, if you look, the human gene, the mouse gene, the rat gene, the dog gene, the chicken gene, whatever gene this is, all of them are the same for this sequence. The frog has one difference right there. The zebrafish, which is a fish, has the green differences, and the pufferfish has these differences. So as you can see, whatever gene this is, all of these organisms have it, and it is very similar in all organisms. As a matter of fact, in the chicken, the dog, the rat, the mouse, and the human, it's exactly the same. DNA profiling identifies an individual by his or her known DNA sequence. One type of DNA profiling involves SNP chips. These are probes that reveal which SNPs are carried by an individual. Again, the SNP chip analysis, we're looking for this particular area. And again, I'd hate to be the guy who has to look at that and find it. And ultimately, it's a computer that does it. No human being, well, the human being that could actually do this would be exceedingly rare. Looking for this particular spot in all of those different genomes. Another method to analyze or to look at genetic material is what are called short tandem repeats. Short tandem, this is a series of A's and C's and T's and G's that repeat. You have a sequence, and then it repeats again, and it repeats again. Maybe not in the exact same spot, but in different places along the gene. So we're looking at a large piece of DNA. I have a short tandem repeat, here, here, and here. Another, a cow has it here, here, and here. But I don't know, let's say a catfish has it here, but not here or here. So we're analyzing and looking at the short tandem repeats. And again, this is a short tandem repeat profile. Again, you see the little peaks are showing, the gray boxes indicate which regions of the individual's DNA were tested. The number of repeats is shown in the box. So here you've got 11 repeats, and here you've got 14, and there's 11 and 13, and there's only 7. So you're looking for a very specific group of bases found in a very particular place. Genetic engineering is a process by which an individual's genome is deliberately modified. A gene may be altered and reinserted into an individual of the same species, or genes from one species may be transferred to another to produce a transgenic organism. Here, in this first case, you take a gene, let's say for instance you have someone who has type 1 diabetes. And we find the gene, the gene is faulty. We take and manipulate, we create a good gene, we insert the good gene in place of the bad gene, and possibly over a period of time, I'll get it out in a minute, that person could be cured of their diabetes because the bad gene that's not producing insulin is slowly being replaced in the pancreas by the good gene, which will produce insulin. Both methods result in a genetically modified organism, or a GMO. So whether we're taking gene out of me and putting it back into me, or we're taking a gene from someone else and putting it back into me, that is a genetically modified organism, or GMO. So, genetically modified organisms are transgenic bacteria and yeast are used in research, medicine, and industry. Bacteria and yeast have been modified to produce medically important proteins. We talked about human insulin, which is produced by transgenic E. coli. Talked about that a few moments ago when we talked about the plasmid. That was the human insulin being produced. Bacteria with fluorescent proteins from jellyfish. Okay, this is a bacteria, and you'll see the bacteria that have the protein. See where they're kind of fluorescing and glowing. So we can do that, and this is one of those experiments you basically do to perfect techniques. Designer plants, methods of inserting genes into plant cells. You have electrical and chemical shock. You have blasting the microscopic DNA coated with microscopic DNA pellets, or bacteria carrying the TI plasmid. Designer plants, we are taking the genetics of one plant and putting it into another plant. Excuse me. Some plants with modified or foreign genes. There are plants that confer insect resistant to corn. There are rice plants that make beta carotene. Beta carotene is the product within certain plants that make vitamin A or help us make vitamin A. Crop resistant and herbicide, or crops resistant to the herbicide glyphosate. Glyphosate is the technical term for a product called Roundup, or it's the chemical term for a product called Roundup. So you have crops that are resistant to Roundup. Roundup is a herbicide. If you take Roundup out and spray it in your flower bed, it will kill everything in your flower bed. But the Monsanto Corporation has created plants, row crops like soybeans and corn and wheat that are resistant to crops. So as the plants start to grow up and the weeds are growing in the old days, farmers were really limited in what they could do to get the weeds out of their crops, because anything they could spray on to kill the weed would also kill the crop. But now they just have one of those crop dusting pilots fly over and blast the field with Roundup, kills all the weeds, but the crops are not resistant. Or crops are resistant, crops are not harmed. So to make a transgenic plant, again, you start with a bacteria that has a plasmid. Here's the gene that we're trying, that's going to impart to this plant whatever we want to do. You know, be drought resistant, be moisture resistant, be resistant to Roundup. We insert it into the plant. We grow it here, we insert it into the plant cell, and we get plants growing and dividing. And the plant is now considered a genetically modified organism. Biotech barnyards, animals that would be impossible to produce by traditional breeding methods are being created by genetic engineering. Transgenic animals are used in research, medicine, or industry. Genetically modified animals also make proteins that have medical and industrial applications. Lots of this early work being done. Some genetically modified organisms, you can see to look at the different corn. This is the normal corn that is being affected by something. You see the kernels are not forming. Looks like they probably have been gotten too wet, whereas this genetically modified corn, there doesn't seem to be a problem with. The debate rages over the ethics of genetically engineering livestock, medical research model, and food crops. Here's the thing we need to understand about our food supply. The human food supply worldwide is controlled by, I think it's 4 companies. There are 4 companies, Monsanto in this country being the big one. And Monsanto is actually all over the world, but it's really big in the United States. 4 companies control our entire food supply. Whether you're a vegan or you like meat, 4 companies are controlling our entire food supply. I could give you some really interesting stories about genetically modified organisms and what happens. In the farming industry, I grew up in South Arkansas. My dad worked for a farmer for 50 years. I know a good little bit about the ins and outs of the farming industry, and genetically modified organisms have really played a key role in the way farming is done today. And it's not always good. Medical research, animals are engineered to carry human disease mutations. Test subjects suffer the same diseases, conditions as humans. Again, lots of genetically modified organisms being used in medical research, trying to make livestock that grow bigger, faster, with less food. All of those things are genetically modified organisms. We could also extend this to human beings. Gene therapy transfers a normal or modified gene into an individual with the goal of treating a genetic defect or disorder. Talked earlier about possibly getting the good gene for making good insulin into type 1 diabetics. That would be an example of this. A normal gene is inserted by ways of lipid clusters of genetically engineered viruses. This is the way the work they're doing over at UAMS. They're trying to use viruses to incorporate good cells. The results are unpredictable. If you insert a virus-injected gene into a chromosome, it can disrupt the normal function and cause cancer. We don't want to try to treat one disease and cause cancer on the other hand. And you could see severe allergic reactions to the vector. So again, working with human beings is still in the very early stages of this type of research. There are some gene therapies being tested as treatments for a variety of human diseases. Again, this is in the early stages, and again, this book was written 4 or 5 years ago, so some of these things may have been abandoned, some of them may have been expanded, but AIDS, muscular dystrophy, sickle cell anemia, hemophilia A. Hemophilia is the disease where your blood does not clot when you get cut. Parkinson's disease, Alzheimer's, various types of cancer, and some other organ disorders can all be treated. There is some early testing of gene therapy. SCID is severe combined immunodeficiency. These are basically children that are born with no immune system. Their bodies don't make B cells and T cells and those kinds of things. Severe X-linked disorder, which means this is a disorder that is most often going to show up in men, but it's transferred to that man by their mother. X-linked traits come from mom, primarily. People affected cannot fight infections and must live in germ-free isolation tents. There was a movie back in the, I want to say, 1970s. John Travolta was in it. It was called The Boy in the Plastic Bubble. There have been some other cases in the media where children were born and they have to live in these sort of sterile cubes. They can't get out in the sun and breathe fresh air because everything in the air would cause them sickness. Acute lymphatic leukemia is typically fatal bone marrow cancer. Gene therapy reinserts modified immune cells back into the patients as treatment. Early tests of this seem to be extremely successful. And again, several years have passed since this book was written, but yes, this is a very one of the go-to treatments now for severe acute lymphatic leukemia is gene therapy. This is a young lad over here, Reese Evans, at age 10. He was born with SCID X1. His immune system has been permanently repaired by gene therapy. Again, put the genes to make the proper B cells and T cells in. He's now making them, should make them for the rest of his life. Young lady over here was just days away from death. Experimental gene therapy for her acute lymphatic leukemia. 2 years later, she remains cancer-free. Again, 4 or 5 year old text. I hope she still is. Should we engineer superhumans with breathtaking strength or intelligence? Insert photo, page 185. There it is. Young lad with his little swim goggles on, his little cape, and his little shorts. That's really, really dangerous right there. They've got that cape knotted around his neck. That's really dangerous. I know we all did it growing up. My mother actually used to use a big safety pin and pin my cape on me when I wanted to be Superman, but knotting it like that, very dangerous. He tried to go down a slide, for instance, and the cape hung at the top of the slide, and he fell over the side of the slide. It would hang him. So, if you got children and they want to play Superman, don't tie that in a knot. Put some Velcro on there, or something that will break away and let it go away. But should we do this? Should we engineer superhumans? No, we've done it in the past. Human beings have done it in the past. Back in the early 1920s, Stalin in Russia wanted to crossbreed human beings and gorillas to produce these big giant super soldiers. So again, that's not the same as what we're talking about here, but whoops, you find some gene in maybe somebody who has a mutation to produce twice the muscle mass as normal. Well, you take that gene out and you try to put it into a whole bunch of people. Should we do that? We find specific genes that make some people more intelligent than others. Should we try to breed a group of people that are more intelligent than everybody else? Again, these are ethical questions. So, genetically modified foods. The arguments against them, are they going to produce more allergic reactions? If the plant that produces the food gets into the natural environment, is it going to push out other plants, more native plants? High profile DNA, criminal case involving DNA. This right here was deeply involved in the O.J. Simpson case. Now, I don't care which side you fall on. He did it, he didn't do it. The DNA and the evidence showed that at the very minimum, he was there. He was at the scene where it happened. Now, did he do it? Did he watch it? Did he stumble on it later? That's all conjecture. But the DNA shows he was there. So why was he acquitted? Because when the prosecution exhibited their DNA evidence, they went into such minute detail that they put the entire jury to sleep. And all the defense had to do is show O.J. trying to put the glove on, and it wouldn't go on. And Johnny Cochran said, if the glove won't fit, you must acquit. But, and then, of course, the prosecution didn't bring in the fact that everybody who has ever owned a pair of leather gloves knows if you get a leather glove wet, when the glove dries, the leather shrinks. And also, if you will watch that trial, which I'm sure you can see it on YouTube, he's trying to put the glove on over a latex exam glove. Try that sometime. Just try to put a glove on that fits, put a latex exam glove on, and try to get that other glove to come on. So again, I don't know whether O.J. did it or not. I know that the DNA evidence shows he was there at some point, but DNA evidence, it was there. It shows whether or not the crime was committed. If you have any questions, please contact me, trussell@uaptc.edu, or the contact button, course messenger link. Again, some of you have still not done the several assignments, the 10 and 15-point assignments in LearnSmart, or I think it's called no, it's not LearnSmart. It's whatever the name of the product is. I apologize. But they're there. Unit 1 materials, open them up, open up the Cengage site, and do those 10 or 15-point assignments. They will make a difference come grade day in May. If you have any questions, contact me, and I will see you next time.